Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. We have come towards the end of this course. So, this lecture number 62 and 63 basically I have focused to tell you something about molecular farming. So, lecture 62 is basically the creation of or generation of transplastomic plants. And molecular farming means you want to produce some pharmaceutically important products in plants. So, normally I have mentioned all these things, but here particularly focus will be either antibody or some hormones. So, in the next class we are going to talk about that, but first in this class we will see that why transplastomic plants. First of all, what is transplastomic plants is basically when you transfer a gene of interest into plastid, and if the gene is integrated with the plastid genome, then it will be possible for you to raise the transplastomic plants. So, gene transfer to plastid, and obviously, what is the advantage, why should we transfer the gene into plastid? Then we will see gene construct, selectable marker genes, plastid transformation vectors, and then expression cassettes. So, these are the concepts we will discuss in this course, and in this class, and the next class we will talk about the application of this knowledge. Okay. So, what you need to do now is that nuclear genome and plastid genome we will compare. So, nuclear genome, one important point is that multiple insertions are common, whereas in the plastid genome specific site by homologous recombination. This may have some advantage over the other. And another point is that in case of transcription, it is basically each gene usually expressed individually. So, that means for each gene you have to put a separate promoter and separate terminator, whereas the plastid genome is basically prokaryotic type. So, it follows more or less a operon concept. So, therefore, a single promoter can control the expression of multiple genes in the form of a operon. So, that is the advantage, that means that multiple genes can be expressed relatively easier way in the plastids. And the current limitation with the nuclear genome is that the level of expression is unpredictable, because where it will integrate, it is a random integration, whether you do vectorless transfer or agrobacterium mediated transfer. So, if it integrated in transcriptional active sites, then the gene will express. If it is integrated in the heterochromatin region, so it will not express. So, these problems will be there, and then there are other problems like gene silencing. So, that can also happen, whereas the difficulty in the plastid transformation is that obtaining homoplasmic. So, homoplasmic term I will explain little bit later. Homoplasmic means in a brief that a plastid contains circular DNA. So, the number may vary from 20 to 100. So, and ideally each and every plastid circular DNA should contain your gene of interest, and if that is happen, then homoplasmy will be achieved. So, that is difficult to obtain, and that is the challenge. Okay. There are some other advantages with the plastid transformation, which is basically maternal inheritance and all other things. Now, we compare the chloroplast and nuclear genetic engineering. Okay. In case of level of gene expression, when you see about the level of gene expression, so its polyploidy results in abundant transgene transcripts and high accumulation of foreign proteins. Why it is so? Because as I said that a single plastid may contains from 20 to 100 circular DNA, and if each circular DNA receive the gene. So, that means each plastid contains, if there is 100 circular DNA, 100 copies of the genes, and then a cell contains say around 50 plastids. So, 50 into 100, that means 5000 copies of genes. So, that means the level of expression will be so high, and you cannot compare with that of the nuclear transformation. And other interesting points I have mentioned, that it is basically the genes are arranged in operons. So, it is possible that polycystonic RNA can be produced and multiple transgenes can be expressed in a single event, whereas in case of nuclear transformation, it is basically monocistronic mRNA, that means each individual genes to be controlled under individual promoter and terminator. And other important point is this, that position effect, that means the plastid does usually the recombination happens through homologous recombination. So, and that eliminates position effects. So, whereas in case of nuclear genome, that is the problem. Now, gene silencing is not at all found in case of plastid transformation, but whereas in case of nuclear transformation there is a possibility of gene silencing. Another most important point is the maternal inheritance, that means the plastid transformation is the transplastomic plants are maternal, that means the pollen does not contain the transgene. So, and that means there is a problem with the transgenic plants about the pollen escape leading to the formation of superweeds. So, such problem can be alleviated if the transplastomic approach can be used. So, that means in nuclear genome it is paternal inheritance. Other important point is this, when you transfer a gene into plastid and if the genes expressed in plastid, then it will produce the target protein, and it has been found that target protein once it produces it will undergo post translational modification such as disulfide bond formation and other folding, leading to the formation of correct bioactive proteins. So, that is possible, whereas in case of nuclear transformation you have to target that particular protein towards endoplasmic reticulum. So, that means a signal peptide sequence to be added, and you have to make sure that the protein 1 goes inside, it should not come out. So, therefore, a certain modification needs to be made in the construct itself, and so the transgene lines are uniform gene expression. So, these are the lot of advantages of the nuclear genome of the chloroplast genetic engineering if you compare with the nuclear genetic engineering. But there is one important point to mention here, that when you transfer a gene into the chloroplast, so chloroplast lies within the cell. So, it has a double membrane structure, and then it is there in the plant cell. So, and then outside the plant cell is basically you have the cell wall. The light portion is this is the plasma membrane, and this is bilayer chloroplast membrane, and this is cell wall. So, when you transfer a gene into plastid, the gene should be able to enter through the cell wall, and then it will also pass through the membrane, then it will pass through the cytosol, and eventually it will enter into the plastid. So, that means a lot of energy is required, and therefore, the particle bombardment method is usually used to transfer the gene of interest into plastid. Now, point is this, that as I said that this plastid contains circular DNA, say for the sake of simplicity I put only one, and this circular DNA will receive the gene. So, here is basically the gene, so it is coming. So, maybe here and here it will get it, but in this case this is basically the case of heteroplasmy. This is the case of heteroplasmy. Now, homoplasmy will be achieved when all the circular DNA of the plastid contains your gene of interest. So, then this will be homoplasmy. So, achieving homoplasmy will be a challenge, and if homoplasmy is achieved, then the level of expression will be very high. So, so next question is that how homoplasmy is achieved. So, how homoplasmy is achieved? So, this is basically achieved through the use of selectable marker gene. So, what is the selectable marker gene? So, the selectable marker gene mostly used is AADA. So, AADA stands for amino glycoside 3 prime adenyl transferase. So, this is basically the AADA gene. So, which is aminoglycoside 3 prime adenosyl transferase. So, what is its function? Its function is basically AADA degrades spectinomycin. So, if you have spectinomycin or spectromycin in the medium and the plant absorbs that, then and if the plant tissue contains AADA, so then this what will happen, this AADA is basically converts this spectinomycin into adenyl spectinomycin. Adenyl spectinomycin. So, normally this spectinomycin is active, but this spectinomycin is basically inactive. That means, if you grow the tissue in presence of spectinomycin, so spectinomycin is basically will bleach the cells. So, that means, it basically bleach all the plastids. So, eventually the tissue will die. Whereas, if you use the plant which contains the AADA gene. So, AADA gene upon expression will make AADA which is amino adenosyl 3 prime adenyl transferase. So, that upon expression will inactivate spectinomycin by transferring into adenyl spectromycin, and that is how the plant will survive. So, so the transplastomic plant should show resistance to this aminoglycosidal type of antibiotics. So, how it is being done? So, I will show it in a another schematic diagram, but say for example, this is the plastid which contains several circular DNA, and it receives the gene 1 or 2. So, this is of course in a tissue, and that tissue is will be incubated in a medium, and that medium contains AADA as antibiotic. So, the selection medium containing then what will happen here in the next case, that only these two will survive, whereas this all will degrade. So, in the next phase what will happen? We will continue to do this selection. So, I will put it here. So, you will be able to achieve a condition where these two plastid, when the cells started dividing. So, these two circular DNA also replicates. So, as a result of that it makes 2. So, now this survived and it contains sorry. So, this contains this gene of interest. So, this in the next round what is going to happen? So, you continue to give AADA selection. So, AADA selection means you have to give the you have to grow with the spectrinomycin. So, better we write spectrinomycin here. So, spectrinomycin So, eventually what will happen? You will be able to achieve a plastid where all these 6 circular DNAs will be produced by the subsequent cell division, and all these will have the gene of interest here. So, this is this condition, first this condition is called heteroplasmy. PLASMY, PLASMY. PLASMY. So, this is heteroplasmy, this is partial homoplasmy, okay, and then this is full homoplasmy. When this is full homoplasmy, then you will have your desired genes also, if you made the construct in such a way. So, the construct contains the antibiotic resistance gene along with the gene of interest. So, all the circular DNA of a single plastid contains the gene of interest along with the antibiotic selectable marker, and in that case all the plastid within the cells will get the gene, and all the tissue will get, all the plastids present in all the cells will get the gene. So, you think that the level of expression will be very high. So, this is how the homoplasmy is achieved. Now, here what is what I am showing here, that is basically telling that is a maternal inheritance, and so therefore, the trait will be restricted with the seeds. So, pollen will not contains any gene of interest, because the disintegrated paternal plastids. So, only the maternal plastids will contain the gene of interest, and here what is showing here, that the multiple gene expression it is possible using the transplastomic plants, and that leading to the formation of biopharmaceuticals apart from the transgenic plant stress resistance. So, now what we are going to see, that this is the method of gene delivery. So, as I said that it is bioelastic gene transfer. So, it requires high energy, so that the DNA that which contains the gene of interest along with the antibiotic resistance marker gene should be able to go through the cell wall, cell membrane, and ultimately it enters into the chloroplast by piercing the chloroplast double membrane. and then so it is again it will be coated with either gold or tungsten, and that will enter into the plastid, and eventually it depends on the homologous regions which is there in the construct and the similar homologous region there in the plastid. So, there will be homologous recombination. So, now let us consider here this point, that is the single gene. So, single gene normally what is to be done, that promoter is there and then you have the marker, marker means it is the antibiotic resistance marker and the terminator. Then you have again the promoter and X is the gene of interest and then the terminator. So, this is how the single genes are transferred, but multiple gene can be transferred under the control of a single promoter, but then and the genes can stack together, but in between you have to put a ribosome binding site, and then the construct is to be made in this way, and then the whole construct can be transferred successfully into the plastid genome, and the single promoter here can take care of the expression of the entire operon. Okay. Now, what I have mentioned is AADA. AADA is basically an antibiotic. So, but there is a it is a negative selection marker, but there is a positive selection marker where betaine aldehyde dehydrogenase can be used. So, where the if the betaine aldehyde dehydrogenase is used, so that will basically degrade the toxic betaine aldehyde. Normally betaine aldehyde is toxic to the plant tissue, but if betaine aldehyde dehydrogenase is there, so that will convert betaine aldehyde dehydrogenase will convert toxic betaine aldehyde to a non-toxic product, which is glycine betaine, and that is utilized by the plant. So, point is this. So, here this is the positive selection marker. So, the glycine betaine is utilized by the plant for its own nutrition, and betaine aldehyde dehydrogenase can be used as a positive selection marker, whereas AADA is basically a negative selection marker. Now, what is showing here is this one is the heteroplasmic diploid. Okay. So, what you see that when the genes are transferred, if you notice out of these there are two different colors. So, this. So, these 4 or 5. So, these plastids are different from the other, that means this has received the gene. So, now what has happened, that through this selection of antibiotic resistance marker, eventually when you notice here all this contains these genes. So, this is a homoplasmic achieved. So, how it is achieved I have explained in the previous slide, and next is this is another pictorial diagram. So, this is how the foreign the tungsten or gold particle containing the gene of interest is inserted, and here you see that it is only one plastid, one DNA of one plastid received the gene. So, that is primary plastid transformation event. Then through round of selection, round of antibiotic selection, what you see that this is a heteroplasmy. Why heteroplasmy? If you notice carefully that out of 4 circular DNA only 2 have the, here out of 4 3 have the things, here out of 4 achieved, but here out of 4 only 1, here out of 4 only 2. So, it is definitely not a homoplasmy, but heteroplasmy. Then further antibiotic selection, eventually if you see all this contains gene. So, this is full homoplasmy achieved. Okay. So, this is the beauty of plastid transformation, and now we will talk about the vectors. So, normally what happens that I said is the homologous recombination. So, there is a region of homology here. So, this is the plastid DNA, which is this left and right targeting regions. So, these are sometimes these inverted repeats are also there. So, and when the vector has to be constructed, one has to take care that there should be a similar sequence to be put here in the vector and another similar sequence of the other to be put here. So, the left targeting region and right targeting region, the similar sequence to be put in the vector where you have the marker and the gene of interest. So, that means where you have the marker and the gene of interest. So, which is clearly showing here and then so this is basically the DNA which is coated with tungsten or gold particle and this will be bombarded into the cell and eventually it enters into the plastid and then a situation will come, these two will meet and as a result of that there will be homologous recombination. So, as a result of homologous recombination, what you will see that these two will fuse and form a single circular DNA where gene of interest and marker both are there. Okay, so, and then transform plastid DNA marker gene can be removed by subsequent selection. There are different techniques for removal. So, that I am not going to discuss. Okay. Now, so this is what I have said is the process. So, first you bombard and then selection, finally regeneration, then additional round of selection, make sure you have it achieved homoplasmy and then you check for the traits or the products. So, this is basically a plastid transformation vector. So, where these are the regions of homology A and B. So, or the flanking regions and here it contains AADA is the marker gene along with a promoter and a terminator. So, untranslated region, it contains AADA gene is tethered to the plasmid promoter and signed Dalgarno sequence. This is signed Dalgarno sequence and a plastid untranslated region UTR which is red. Okay. And then homologous recombination and this is the plastid DNA and this is the vector DNA. So, upon homologous recombination, so this AADA will be inserted or integrated within the plastid DNA and so basically it happens by physical linking to the AADA gene by the flanking region and then eventually you will be able to have this AADA in all the circular DNA of the single plastid and eventually in all the plastid DNA present in the cell. So, the transgenic cassette which need to be constructed should consist of a promoter signed Dalgarno sequence 1, then you should have the coding region 1, then again signed Dalgarno sequence 2 and then you have the coding region 2 UTR, like that way the genes to be constructed. So, here what I am showing, this is typically a vector. So, here the this AADA gene is inserted, but apart from that there is a multiple cloning site here where the target gene can be inserted. So, we will not go into that details and here there is another thing is this that in order to modulate the expression sometimes the translation control region TCR region is to be added. This is 5 prime TCR. So, that will basically modulate a stronger expression than the one. So, the TCR, so this means that here you have the promoter plus 5 prime UCR and this is basically in addition to that you have a translation control region. So, so this is there is a copy paste pattern. So, this is basically 5 prime or 0. So, a PL cassette encodes the promoter 5' untranslated region 5' UTR and or B a 5' translation control region 5' TCR, which means 5' UTR plus a segment of the coding region. So, that is the difference. So, there are basically way the constructs can be made, so that you can get a better expression. So, that is just for your information, and then these are the different ways transformation can be done. Okay. One is that suppose if you want to do a insertional mutagenesis. So, that can be done where what will happen, the existing gene will be disrupted in this way, and the other one is that if you want to put the gene of interest in the plastid, a new gene by replacing the existing gene, that is also possible and so or a point mutation can be introduced, or simply you put the gene which is not there in the plastid. So, that can also be integrated, and so that the plastid gene DNA will receive this. So, this will be more clear here. So, here what you see that left flanking region, right flanking region, and this is the plastid DNA, this is the vector. So, gene of interest is here under the control of a promoter terminator, and AADA is here, and then that is inserted, and eventually you will get the whole thing here. So, and then what happens now, the difference between the left and right part is that here the one single plastid is here, and then one single terminator is here, but both the genes are joined with the signed Dalgarno sequence and interspacer element. So, that this plastid will do this promoter will basically transcribe both the genes. So, that leads to polycystonic mRNA, and then eventually there will be cleavage, and that makes two separate mRNA. And this is at the last that if you want to have optimize, if you want to optimize plastid transgene expression in non-grain tissue. So, that is possible by doing certain fusion. So, one is that if the RNA extracted from the green tissue as well as from the ripened tissue, and then what will happen, that this transcriptomics leads to the giving the transcriptional signals. So, which promoters and which promoters which is active in the chromoplast. So, that promoter to be inserted here, and along with that the polysome can be isolated, and then that can be translated. So, translatomics can be added. From there translation 5' UTR signals can be isolated, and that can be fused here. So, as a result of that, chimeric expression element, one coming from the unripened tissue, one coming from the ripened tissue, and that to be fused and that will be put in the upstream of the gene of interest, and that leads to eventually more efficient expression of the genes into the plastid. Because of the shortage of time, and because I am not supposed to discuss about these techniques in this class, because our aim is basically look on the pharmacognosy and the molecular farming. So, with briefly by introducing this, I will end this class. So, by this you have developed some understanding that how genes are transferred into the plastid and what are the advantages if genes are transferred into the plastid. So, that means that there will be huge level of expression, and there are lot of advantages, that is maternal inheritance, no chances of pollen escape, the level of expression will be high, and there will be no gene silencing, you can have the predicted expression. So, this all speaks positive or advantageous over the nuclear transformation techniques. So, in the next class, which will be the last class of this course, we will talk about molecular farming, where human somatotropin gene will be expressed in tobacco using this transplastomic techniques. With this, I end this class. Thank you.